Screams louder.
Hello guys and welcome back to Biology Insights. We are here with another video lecture of the series Inheritance Biology. In previous video lecture, we dug the history of developing the chromosome theory of inheritance, which disclosed immense work of many great scientists played an important role. We saw how Sutton and Boveri strengthened the connection between chromosome and hereditary, but yet concrete proof for it was to be given. A gene which is linked to chromosome was required to examine to understand chromosomes' behavior during reproduction. All these requirements were fulfilled by an American biologist, Thomas Hunt Morgan, who discovered a particular eye color mutation in fruit fly Drosophila melanogaster. Later, his student Calvin B. Bridges obtained definite proof for the chromosome theory of inheritance. In 1909, Morgan performed experiments on mutant male fly that had white eyes instead of red eyes of wild-type flies. When this male was crossed with normal eye-colored wild-type female, all the progeny had red eyes, which indicates that red was dominant to white. When these progenies were intercrossed with each other, Morgan observed that interestingly all the daughters had red eyes. But in male progeny, only half of the sons had red eyes, the other half had white eyes. Based on this pattern, Morgan proposed that the gene of eye color is on X chromosome, not on Y. So the white eye-colored male are having X chromosome with mutant gene, and because they have only one copy of X chromosome, they are affected by the gene. While in case of females, even if one of the X chromosomes is having mutant gene, as red is dominant over white, they do not express it and only be an unaffected carrier. Morgan carried additional experiments to confirm the elements of his hypothesis. In one experiment, he crossed a heterozygous female with hemizygous mutant male, where word hemizygous proposed that the individual is having only one copy of gene. In another one, he crossed homozygous mutant female to hemizygous wild-type male. In both the cases, he got expected results with half the progeny having red eye and other half having white eye. Thus, Morgan's hypothesis of linkage of eye color with X chromosome was proven to be accurate. Later on, Calvin B. Bridges, student of Morgan, performed the experiment on large scale to secure the proof of chromosome theory, which eventually opened a new perspective to the theory. He started the test by crossing white-eyed female with red-eyed males, and similar to Morgan's, he observed either red-eyed females or white-eyed males. But an interesting deviated observation was exceptional flies, who were having white-eyed female and red-eyed male. He tried to cross these exceptional flies, but he observed that the female was fertile but the male was sterile. Now the question was, what caused these exceptional progenies to arise? Is there any unique event occurring in these which made them exceptional? Well, thanks to Bridges, who gave really satisfactory answer to it. He proposed that the exceptional F1 flies were result of abnormal X chromosome behavior during meiosis in the female of parental generation. In normal cases, the X chromosome in female would be disjoint or separate from each other during meiosis, and when they are fertilized by normal sperm, they give expected progenies. If they fail to separate, they create an egg with either two chromosomes called diplo X egg or no chromosomes at all called nullo X egg. Fertilization of such abnormal eggs with normal sperm would produce zygotes having abnormal number of sex chromosomes. So if we consider a sperm with Y chromosome, the progeny would be either XXY or YO, where O represents absence of chromosome. Here XXY shows white-eyed female and YO did not survive at all, which proved importance of X chromosome in viability. If we consider a sperm with X chromosome, the progeny would be either XXX or XO. The XO was red-eyed male and always sterile, which proved that in the case of Drosophila, Y chromosome has nothing to do with the sex determination, but must be important for male sexual functions. The XXX zygote developed into red-eyed meta-females, which can be distinguished from normal females by anatomical abnormalities, including ragged wings and edged abdomens. These are called super females, though there is no such super about these females. Poor ladies. Bridges called this abnormal chromosome behavior non-disjunction because it's failure of chromosome to disjoin during meiosis. Though he was not able to explain the exact cause behind it, he had greatly strengthened the view that all the genes are located on chromosomes, and Mendel's principle could be explained by the transmission properties of chromosome during reproduction. This is called Chromosome Theory of Inheritance, which is the most important achievement of biology. It is often difficult to distinguish what Morgan's work and what is that of his students, but nobody has ever doubted that Morgan was the ingenious leader. And therefore, in 1933, Thomas Hunt Morgan was awarded the Nobel Prize in Physiology or Medicine for his discovery concerning the role played by chromosome in heredity. The finding that genes are located on chromosomes made it possible to explain Mendelian principles in terms of meiotic behavior of chromosomes. Let's learn them in detail. The first one is principle of segregation. We know how Mendel established it, as the allele of single gene segregate from each other. Connecting it with chromosomes, during meiosis, the homologous chromosomes, one from mother and one from father, pair with each other. Recalling Drosophila example, let's consider the mother chromosome has dominant allele and father chromosome has recessive allele for eye color. In the anaphase of first meiotic division, the paired chromosomes separate and move to opposite pole of the cell. One carries dominant allele and other carries recessive allele, which eventually resides in different daughter cells. Mendel's principle of segregation is therefore based on separation of homologous chromosomes during the anaphase of first meiotic division. The second one is the principle of independent assortment. Mendel said that the alleles of two different genes assort independently. To understand the relationship in case of chromosome, let's consider two genes that are on different chromosomes, one for eye color and one for body color. During meiosis 1, homologous maternal and paternal chromosomes pair with each other. As there are two pairs of chromosomes, two different metaphase alignments are possible. During anaphase, when disjunction occurs, there is 50% chance that the dominant allele of both the genes will move together to the same pole, and a 50% chance that they will move to opposite poles. Similar situation is for recessive alleles as well. At the end of the meiosis, when chromosome number is finally reduced, in case of first alignment, gametes will contain either dominant or recessive combination of alleles, while in second alignment, gametes will contain new combination of dominant and recessive alleles together. Mendel's principle of independent assortment is therefore a statement about the random alignment of different pairs of chromosomes at metaphase. So, till now we have learned about Mendelian principles and how they are followed by different means, even by chromosomes. But do you think each and every gene present in all organisms living on planet is following Mendelism? If not, then what are the other scenarios? We will see the exceptions of Mendelism in the upcoming video lecture. If you enjoyed the lecture, don't forget to give us a thumbs up, subscribe to the channel and hit the bell icon so you will be notifying whenever the new insights of biology are introduced. Thank you for watching. See you in the next lecture.